Welcome to BioChat, a podcast by ABclonal Technology. My name is Ken Lung, and with this podcast, we aim to familiarize you with not only ABclonal's contributions to efforts in scientific discovery, but also to highlight the direction of ongoing research and help scientists determine how to best leverage their skills to improve global human health and quality of life. Join me today in welcoming Dr. Hai Wu. He has a PhD and is the Chief Technology Officer at the Antibody Division at ABclonal Technology. Dr. Wu co-developed the single B-cell-based monoclonal antibody discovery technology known as SMab. This enables high-throughput development of high-affinity, high-specificity monoclonal antibodies, and the resulting antibodies significantly surpasses those developed using traditional technologies such as hybridoma and phage split. Antibody products generated using SMab have been widely adopted as research tools, diagnostic products, and therapeutic leads. Prior to joining ABclonal, Dr. Wu earned his PhD in genetics from the University of Rochester and completed his postdoctoral training at UMass Medical School. Thank you for joining us today, Dr. Wu. How are you doing?
Thank you, Ken. I'm doing well. I'm very glad to have this opportunity to speak to our audience.
Yeah, so I always ask my guests this, and we are colleagues at the same company, but why don't you tell us why you love science, why you ended up doing what you do now?
I think loving science has originated from my curiosity to the natural world. I grew up in a rural area, and as a kid, I always did a lot of, I would say, exploratory activities like, catching little fish, shrimp, and, playing little insects. It's just inspiring, and it's kind of a natural. And with the curiosity growing up, I end up more towards biology major, and eventually end up with a PhD degree in genetics.
So how did you get from genetics to antibodies? That's kind of a curiosity, because usually with genetics, you work with DNA, but based on your genetics background, you would probably have a lot of very relevant experience in engineering antibodies from their genes, from the IgH and IgL genes.
I think one of the beauty of doing science is that sometimes you're doing one topic and you bump into another topic. So my initial interest into antibody is when I started genomics and epigenetics. We use a lot of antibodies to do ChIP-seq. We are suffering from getting good antibodies with consistent performances. That's really a bugging issue. Even with some of those end products from leading providers, sometimes you have all these batch differences. That's one of the driving force for me to develop good antibodies. When I have a chance to co-develop this single B-cell platform with Dr. Yang Lo at Eurogen, and this is one of the main reasons that brought me into the antibody field.
That actually gives us a perfect segue into our main talk today, which is the advantages of ABclonal's SMab platform. So you work for the custom reagent services, particularly the antibody production pipeline, where we are going to provide optimized antibodies that are highly specific, that bind very well. They have high affinity. They have broad applications in whatever experimental application that they need, because they have a particular target that doesn't have a commercially available antibody, like you said. We can take this opportunity to educate our audience about SMab.
Yeah, SMab platform actually gets its name on that uses single B cell, and we want to develop a superior antibody to our client. And this platform is a way that you culture B cells, and we think this is one of the best technical route that could save our client cost and give us the best result. Briefly, I'll describe the process, how SMab platform works in our pipeline. So first of all, we would get high quality antigen and immunize the animal, mainly in our cases, rabbit, and we would isolate either PBMC or splenocytes from the immunized animal. The cells will go through enrichment and sorting, and those B cells that recognize the antigen that we're interested in will be sorted out in a single cell format into each well and cultured for extended time. In the end of the culturing time, we would characterize the antibody secreted into the cell medium. We use a lot of assays, like ELISA, WB, FACS, or even blocking assays, train ligand and receptor, and to single out those antibody clones that are really fitting to our need. And the rest of them will be saved for later. In a single campaign, we would have FACS grade antibody, or we would also isolate ELISA grade antibody. They're suitable for different applications. So once we have decided a short list of antibodies, we would do traditional PCR cloning to get the HBV gene out from a single B cell expanded clone. And those clones will be transfected into mammalian cells and recombinantly expressed. And using the supernatant from those transfected cells, so we would do a secondary screening, or we typically call it refining screening. So once this refining screening is done, we would end up even a shorter list with antibodies more suitable to our application or applications. So then we would convert them into recombinant expression using plasmid to purify them out and do more thorough validation, either qualitatively or quantitatively. And some of them will also be engineered for special applications. So once these pre-production steps, characterization and validation engineering is done, we would go through mass production. Depends on the need. The final product will be either for our client, or we would put them into our product inventory. Our current focus is rabbit because it has a superior affinity, specificity, but we also can deal with cells from human, other animals like mouse, monkey, and pig, and also llama. It's very universal.
Once you get the genes, you can actually engineer the antibody, but in order to get the antibody in the first place, we don't really have much of a choice because the host immune system of whichever antibody that we use is much more capable of generating that antibody diversity that you can eventually enrich from. So we unfortunately can't get away from animals in this production process.
To me, with years of experience, and also from different conversations with our client and our own product development experience, I think the best antibody typically coming out from those more natural, I would say. We're still apprentices of learning from nature. My first choice that immunizing animal with their good quality antigen to get the best candidate antibody.
But essentially, once you get that antibody and you are able to enrich it through the various culturing and screening processes, you have the gene. You can put it in any cell system that you want to express it in. You can even manipulate the gene to essentially artificially improve the affinity and the specificity of that antibody. Is that correct?
Yes, with or recombinant expression with clear, verified, validate VHVL sequences, you can do all sorts of things, such as FC swapping in humanization, affinity improvement, maturation, right? All sorts of things that you typically see in refining the performance of antibody.
About the culturing, because a lot of different companies use a very similar technique, but ours is unique in that we use an ex vivo culturing system. Could you elaborate on that?
I think we picked this technique with the belief that we also heard from ourselves and also from our clients' experience that they want a more targeted screening. And with that, you want to know the function of that specific candidate clone as early as possible. The best way to do that is in a more nurturing yet stimulating environment that B cell can secrete enough antibodies for those assays. But certainly we've already refined our assays and reduced the needed antibody, make them more suitable for micro volume assays, and yet with good enough precision and reproducibility. And those are a very unique angle and also solution that we provide for the community, that we culture and stimulate the cells in vitro. So that way we would have better and yet faster characterization as early as possible. And also in a way that because you're working with a smaller number of clones afterwards, those clones selected from a bigger pool, right? So you will save a cost upfront. You don't have to randomly, in a large scale manner, deal with a large number of workload, and that also speed up deliveries.
You had intimated that we can do a lot of the potential assay development and assay validation for the customer as we produce the antibody for them. Most of the customers probably use it for Western blotting, which is one of the primary uses of antibody reagents. Some of them will likely use it for IHC or immunofluorescence. Some of them will most likely use it for chip and maybe cut and tag, other dot blots. For things that we normally don't do, how do we accommodate the customer? Do we just give the antibody to the customer and let them give it a try? Or do we develop the assay with them side by side? For example, if they want to do a chip, we would actually get some of their samples and do it within their particular system.
I think we do have the capability of doing that. We have a big team that is technically capable, but we tend to ask our clients to assist us because they know their target better, they know their assay better. For those validations, the quality of samples are very key to the success. And also, in the early on, we would, in the culture, in the primary screen stage, we typically don't go for like check or cut tag, those large quantity or purified antibody fits better scenario. And those will be moved a little bit later to purified antibody stage. But certainly we can collaborate with the client to offer those assay development.
Obviously, the customer can look on our website at ABclonal.com to look for the standard package, request a quote. Afterwards, like if the customer wanted to potentially humanize this antibody or adjust the application, whatever they want, like because you have the genes, again, you can essentially do whatever you want. And because this gene is probably quality assured, so you sequence it often, you make sure that there are no random mutations during the gene construction process. And so you can have a perpetual reagent. So the client could essentially use up the stock of antibody that you've produced, or they can actually get the gene from you and cultivate it themselves at some point, correct?
Yes. And also we can do custom production for them as well. So
that's really awesome. Like there's a lot of versatility with this process. And I see just from my help with marketing these services and talking to you that there's a lot of bang to your buck with this service. And it's actually really, really impressive. I've used antibodies quite a bit. Like they are from an experimental standpoint, from me being in graduate school and using lots and lots of antibodies, the cost does add up. So being able to defray some of that cost through the technologies that you've described is probably very attractive to our customers. And I commend you for developing this platform. This is slated to drop in September. September is actually Blood and Childhood Cancer Awareness Months. Mm-hmm. And we can actually talk about some of the products that we have done with clients to assist with this awareness. For example, we have this case study, a publication in Cancer Cell published in 2019 from the Furnari lab at the University of California at San Diego. And what we have is a study. This is the inhibition of nuclear P10 tyrosine phosphorylation enhances glioma radiation sensitivity through attenuated DNA repair. And they actually had a couple of custom-made antibodies. One of them was from us, from ABclonal. And this is an anti-P10 antibody, but specifically an anti-phosphotyrosine 240 antibody. P10 being a classical tumor suppressor, they are able to use this antibody very well in their publication. And obviously, Cancer Cell is a high-tier publication, not that, something that's in some other journal isn't important, but considering the tier of journal and the quality of their data, I thought maybe we can talk a little bit about the development process for this antibody and how it benefited our customer.
Actually, rapid monoclonal antibody are especially good at recognizing those small changes in epitope. For example, they recognize unphosphorylated epitope, but yet you can develop a specific antibody that recognizes the phosphorylated epitope. For example, this phosphorylated tyrosine 240 and P10. So when we did the project, first of all, we would see there are some differences in recognizing phosphorylated and non-phosphorylated peptide. And the serum we pick coming from a rabbit with a very good titer, and we isolated splenocytes from the animal and put them into cell sorting using the phosphorylated tyrosine peptide. And we ended with 96 different clones. We picked 12 after discussion with our client. I would say actually we have more than two showcased here that works well. And then these two are the best two we showcased here. And the client picked those two as their final delivery. And that worked well to strengthen their studies, which published in Cancer Cell. And we're very happy for our clients that our antibody did help them to elucidate some of the mechanisms that can, in the future, develop either therapies or diagnosis methods or understanding the fundamental mechanisms that will give us a more thorough explanation of cancer progression.
And in this study, based on their results, they were able to show that usually this is phosphorylated by FGFR, which is the fibroblast growth factor receptor at this tyrosine site. But if you can inhibit that, you now sensitize these particular cancer cells to radiation therapy. And that's potentially really important for, the prognosis of these clients. So at some point, a lot of cancers do become resistant to therapy. If you can remove some of this resistance, now you've improved the quality of life and the survival outcome for these patients. So, yay, ABclonal.
Yeah. Actually, a little bit offside topic on that is that nowadays people are thinking delivery those antibodies intracellular that can target those intracellular targets, which previously was not druggable because most of the drug in the antibody drug right now we develop are targeting membrane proteins, receptors, ligands, right? People are developing those intracellular delivery method to expand our target selection, which I would say in the future would fundamentally change some of those previous uncurable diseases.
When you talk about intracellular development systems, I was reading up on camelid antibodies, which can be turned into nanobodies. And so we have this particular case study where you actually immunized an alpaca, of all things, with a SARS-CoV-2 spike S1 protein. So normally when we talk about immunization, we think of rabbits and mice and guinea pigs and maybe chickens. But when you decide to now use an alpaca, what was the basis behind using the alpaca? And I believe that has something to do with the way their antibodies are structured.
Yeah, actually, llama recently, including, the alpaca is a smaller camelids, and with the llama, all these are the camelids in the New World, and they have a very special biology that they would have those single chain antibody only with comprised of two heavy chain, and they have an extra long CH3 domain that would recognize the antigen. And this gives quite advantages in many applications. If the antibody is small, that's why it gets named nanobody, right? It's only about 15 kD without any tag, and is yet functional in recognizing the antigens. I think the other very special part, because it's a small size, you would get into those so-called cryptic epitopes that previously with a larger antibody is not accessible. And those antibody can also somehow be very stable. There are a few studies also showed that nanobody could withstand the challenge of high temperature as high as 90 degrees of heat for like 10 minutes. I remember that a while ago I read a paper that there is a company developed a caffeine-specific VHH antibody to test how much caffeine is in a cup of coffee, which also showcase the stability of nanobodies. And recently, it gets movement in diagnosis and therapeutics because it's small size, easy to engineer. You don't have to deal with the traditional H2L2 pairing issue, right? So it's very attractive. I think there are many other aspects I didn't really elaborate on, but this is a very attractive class of antibody that has a lot of potential in diagnosis, in antibody tool development, including therapeutics.
It just kind of surprised me because when I looked at the structure of a VHH antibody versus the normal, like heavy and light chain antibody, essentially you are missing three CDR or complementarity determining regions because you don't have that second chain. But at the same time, you don't lose all that much in specificity. Is that correct?
Yeah, you don't need that pairing, so it acts by its own.
That's wonderful. And you also alluded to the fact that they are much more like thermostable and they are very robust antibodies, I assume, because, camels need to survive in the heat. So at some point they need to thermoregulate and make sure that their antibodies don't melt or something inside their bodies while they're traveling the desert. But yeah, let's take a look at this SARS-CoV-2 spike S1 protein antibody. So first of all, when you immunize the alpaca, you don't just have the VHH antibodies, you have everything else. So you have to select for the VHH versus the normal IgG complement.
Yes, actually, this is a great question. We always end up with the challenges that how can we differentiate those VHH B cells from IgG B cells. Typically, we would test the total IgG to see if the animal responds properly to the antigen. Yet, we want to know how is the success rate we can get when we check on the VHH specific titer, because that's really meaningful. Not every B cell is secreting VHH. So if we have good titer, that means in the population of B cell, we would have enough B cells that's secreting VHH antibodies. And that will give us a very promising indicators this project work towards the right direction. So in this case, we do see a good titer against the VHH at the second bleed. We used a single B cell platform, isolated VHH specific B cell clones. 896 well-placed culture, we got 38 positive, 26 of them are binding to the receptor binding domain of SMab protein. So, and the diversity is pretty good as well. VHH positive B cells are generally less abundant.
And it's actually really cool that you can differentiate between things that just bind the parts of the spike S1 that aren't as relevant to the study versus the one that do bind to the RBD of the spike protein. Now that you have selected your appropriate clones, you are now able to clone said antibody into an expression vector and reproduce it. And so you did a pseudovirus neutralization assay. Could you elaborate on that?
Yes, so the antibody we developed do have the capability that could neutralize infection virus. So we compared one of the best positive control antibody, which is known that neutralizing its traditional H2L2 type antibody. And we see that a VHH has slightly weaker neutralization assay activity, but it actually can neutralize infection. We did extend the immunization stage longer, and we get even better. VHH could have similar neutralization activity comparing to positive controls.
And because these are larger animals, you can potentially do that because with a rabbit, at some point, they're just unfortunately going to bleed to death, despite however well you are able to treat them via regulations governing animal husbandry. But with an alpaca or a camel, like they're just so much larger that you can potentially do like 3 or 4 rounds. And we did see that, the multiple rounds of immunization do improve the binding of the antibody against your antigen. So that's actually really fun to see.
There were some studies showing that the VHH generation is lagging behind the regular IgG generation. So with the extended immunization time, you certainly get more. But of course, there are some exceptions. Well, overall, with extended immunization, you certainly will get better results.
So these are our custom projects, and what you had alluded to before is that some of the technology that we use, we are actually using to provide reagents within our catalog. So the next thing we can talk about are catalog products via ABclonal that you can peruse at abclonal.com that are available thanks to the SMab technology that Dr. Wu was talking about. So we have ABflow, which is for flow cytometry. We have PolyMab, which is essentially a multi-epitope targeted antibody cocktail. These are all recombinant rabbit monoclonal antibodies, by the way. And we also have PathoQ, which are specifically designed for pathological analysis. ABflow is a directly conjugated primary antibody that will recognize the target on the cell surface or perhaps intracellularly. And the tag is a fluorophore that standard, it comes in 488 nanometers, which is green, and 647 nanometers, which is red, because a lot of flow cytometers in flow cytometer like core facilities have those standard channels. That will allow you to have versatility to just say, I want a blue channel, I want a purple channel, I want yellow or whatever to study some other marker. But for the very standard markers, for example, if you were studying a T cell or B cell biology, for T cells, you would want to differentiate between a CD4 T cell or a CD8 T cell, and you have to use an antibody to gate for that. So what ABflow is providing for our customers is a way to say, you don't have to use a secondary antibody for this, just label your CD4, and now you can do whatever else you want with the panel of antibodies based on however many channels are available on your flow cytometer. So here we have our ABflow 488 rabbit anti-human / anti-monkey CD4.
We developed this ABflow pipelines is that because the affinity and the specificity of rapid monoclonal antibodies are great for those sensitive yet require a highly precision and specificity assays. In this case, flow cytometry, using our SMab platform to develop those antibodies. These specific CD4 antibodies developed from human CD4 recombinant protein reproduced in-house. And we immunize the rabbit, we get good titer, and then we select the best candidate. We validated thoroughly with 3 rounds of experiments at the B cell culturing stage, and then at our later on refined screen called a secondary screen stage. And then we reassure our result using purified antibody. All these antibodies perform very consistently. And with these results, we've already compared with our leading competitors, and our antibodies does outperform competitors' intensity and the narrow peak, which means it stands very well. We have more data coming using traditional sample type like PBMC. And this is one of the core TBNK panel that we are developing. And luckily, this antibody also cross-reacted to monkey. And monkey has been a very important experimental animals used for those drug development clinical stage. They are preclinical stage. They were used as large animals to evaluate the drugs. And we could use those CD4 antibody to monitor the health status or the physiological changes inside monkey body before and after drug administration. And those are great tools to monitor the dynamics of T cells.
I had talked to Rosa before on our podcast about like the differences in antigen design for something that's primarily for a Western blot, but versus something that has to look at a 3-dimensional native epitope, so that you can't like just bomb the cell with SDS and use the same antigen. You have to use a natural antigen, and the fact that you are able to get such good separation between your negative control and positive control is really impressive. We can also talk about B cells. So In addition to T cell markers, we have B cell markers such as our ABflow 647 rabbit anti-human, also anti-monkey, CD19 monoclonal antibodies. This is again a rabbit monoclonal antibody, recombinant, expressed using the SMab technology. And this time it's in the red channel. And perhaps you can tell us a little bit about this one too.
Yes, CD19 has been a very important marker for B cells, particularly those late-stage mature B cells, right? And it's a key marker for B cell to study B cell dynamics in vivo. In this case, we used recombinant and bioactive CD19 that revalidates bioactivity before we immunize the rabbit. The antibody resulting from this project can recognize the two frequently seen cell lines coming from B cell lymphoma, Raju cells and Dotty cells. We also validate the antibody that recognizes the monkey CD19 because human and monkey CD19s are very homologous. They pretty much identical in many of the 3D structures and the primary sequences. So I think this is also a very key tool that we want to bring to our clients that they could use in biotech and biopharma industries for many of those asset development, such as you monitor the B cell dynamics. And we also have other B cell markers like CD20 coming up soon. All these will form nice panel to characterize those lymphocyte subpopulations, which is the key to understand the biology.
And the fact that this panel is ever expanding and you're using, again, this technology that guarantees as much as we can with modern technology, but it guarantees a high specificity, high affinity antibody that will always be available as long as we have the gene, as long as we have the capability of expressing it. So that's ABflow for you. That's a really wonderful reagent, and I hope it makes a lot of our customers' lives a lot easier. This being Cancer Awareness Month, so this is both blood cancer and childhood cancer awareness month, we thought we'd take a look at some cancer-specific antibody targets. So here we're actually talking about our polymab antibodies. How do we choose the epitopes that go into the cocktail that makes up these recombinant rabbit monoclonal antibodies that recognize this particular target? Like, what is the thought process behind that?
The level philosophical thinking is that we always come to the client and get the feedback that some of the antibody only fits to one species or one certain application. They want an antibody product that applies to different applications, right? In this case, IF or IHC, and also can do good quality WB pictures for publication. And we learn from the nature. So polyclonal antibody has been existed for 100 years, over 100 years. But the beauty of polymab is that we make it more reproducible because we have all these defined clone of antibody, monoclonal antibodies, being mixed carefully that they recognize different epitopes. And those epitopes typically could be adjacent to each other with a decent distance without interfering on the binding of two antibodies. Right? So the antibody binds pretty much independently. Sometimes one antibody binding could facilitate the other antibody's binding. So that would stabilize the structure, local fragment. So that would sometimes generate new applications. In this case, we have two linear epitopes that recognized by two antibodies in either mouse or either human. But once we combine them together, they have a synergy that would stabilize their binding on the cell and can perform IF application here. So that would expand the antibody into new applications. So we have more coming that was reminiscent to the polyclonal antibodies multi-application nature, yet with the quality, stability, reproducibility of monoclonal antibody. And so we just try to combine the beauty of both into this special product line.
Yeah, that's the best of both worlds when you have something that has all those different applications, but you know that the previous rabbit died, we got to immunize a new rabbit. This antibody doesn't work as well, but because you specifically engineered this antibody and this antibody can be reproduced recombinantly, you basically have this antibody for as long as the Earth exists and DNA exists, right?
Yeah,
In this case, CDKN1A is the P21 molecule. It's under the control of P53, our little cancer connection here. Let's talk a little bit about cathode-Q. So in this case, we're talking about immunohistochemistry with both fresh sections and also paraffin-embedded formalin-fixed sections. So in this case, we have our beta-catenin rabbit monoclonal antibody. It's also knockout validated. So there is data on our website showing that when you knock out the gene, there is no band. So that's like the easiest and probably most visual way we can confirm that this is in fact specific. So as you know, beta-catenin acts downstream of canonical Wnt signaling. It regulates cell adhesion. It's associated with a lot of cancers and especially colorectal cancer. Perhaps you could tell us a little bit about how the development process for IHC-based antibodies is performed at ABclonal.
Q product lines are the premium IHC grade antibody product lines that we are growing. The process is a very thorough validation as the key to develop those high quality antibodies. From the beginning to the end, that we use different types of IHCs. For example, in B cell stage, once we have B cell culture supernatant, we have those very mini size, we call it a mini IHC setting that we could use only small amount of B cell supernatant to cover tissue microarray that would help us to select the IHC grade antibody from the beginning. I think IHC has been a little bit hard because we know sometimes people say, oh, WB grade antibody is good for IHC, and they are only candidate. If you really want to test their performance, you have to use the real application. So we start in the correct way, screening the antibody using IHC itself to test its performance in IHC. And after that, we have those recombinant supernatant and purified antibody. They will go through from mini IHC to multi-sample TMA slides to thoroughly validate and evaluate its performance. We use both certain number of positive controls and also negative controls to make sure they have good signal in positive samples and very low or no background binding in negative samples. Beta-catenin, certainly, as you said, is a key protein and transcription factor in Wnt signaling. Wnt signaling involves almost every aspect of cell physiology and pathology.
And this is one that you had told me about. This is actually MSH6, also KO validated. This is a DNA mismatch repair protein, and the disruption of DNA mismatch repair obviously means you accumulate mutations, and now you have all kinds of cancers that are possible, including colorectal cancers. So what we have here that our audio listeners can't see, but those of you who are watching on YouTube can. We have a Western blot of various cell lines, and we also have the KO experiment where you can see definitely, like, if you've knocked out this gene, there is no band. And now we also have an IHC image of both the human appendix and of the human colon carcinoma, and you can see just this beautiful dark brown where you have positive staining. So what can you tell us about this one, since you seem very happy about this one, Dr. Wu?
Yes, I'm very happy about this because if you look, certainly you have a very specific antibody that recognizes MSH6. We have an in-house antigen designing team, very capable, that also we get ideas from the structural data from protein 3D structures, that we specifically select a small peptide that acts, that's accessible molecule's surface also thanks to the many years of structural biology study, we have this data available. And we chose this specific peptide, which has very good specificity. It has least homology to MSH and related proteins, very unique in this MSH6 protein. So that's one of the fundamental aspects we bring it to the wellness of our client, that we have to get a proper, good antigen design before we develop antibodies. So this carefully chosen peptide did reward it as well, that we not only get good WB antibody, but yet we have this beautiful, clear, strong signal, and the subcellular localization is clear into the nuclei here, that which is where this protein resides in. It involves in the DNA mismatch. It stays in nucleus. The cancer area, you see the great, strong, dark brownish staining pattern. Yeah, but certainly some of those proteins would reside in, the protein would appear in those normal cells, but it just has a stronger signal in cancer-related tissues. It's a great antibody. So I would say it's a good antibody is also coming from a carefully designed project, from the antigen design, screening plan that we used for identifying those antibodies, and thorough validation. And of course, in the end, we would have reproducible and highly qualified production team to deliver consistent antibody products.
Yeah, these are all hands on deck type products, and it speaks to the conceptualization, the experience that you all have with this platform and with just generating high quality antibodies from the first time you think about it on a computer all the way to the last drop that comes out of the affinity purification column. So this is actually a really wonderful project.
I would say SMab platform has given us very critical tools and yet expandable platforms that to develop quality products. At the same time, we will also offer these wonderful tools as our CRO platform to benefit a broader community. It's not, our product line team cannot develop all products to cover all targets. I think those CRO projects are more tailored into our clients' needs, that they could develop their own products. And that's a wonderful platform. And also, I would advertising a little more, to our product lines. Our team has been spent almost 3 years to develop those high-quality rapid monoclonal antibodies. Some of them, as you've seen already here, in the different product lines, really fit into different specialized applications. Yet we have more antibodies, high-quality monoclonal antibodies from rapid product development stages, different stages. They are coming up in the coming years, and we hope our efforts, antibody development, could bring more good antibodies, yet with affordable price that would prepare our scientific community better into the future.
And with the versatility afforded by this platform, I think there's a lot of potential for the future, and I'm very excited to see what comes out. Dr. Wu, thank you so much for hanging out with me today. I really appreciate your time. Best wishes to the continued development of SMab, and I hope we have a lot more customers to serve in the future so that we can really work together to bring up positive change to human health and quality of life.
Thank you. Thank you, our audience.
All right, this has been a conversation with Dr. Hai Wu. He is our Chief Technology Officer at the Antibody Division at ABclonal Technology. We hope you will join us again next time when we will explore another exciting topic about bioscience research, biotechnology, and careers in adjacent to biosciences. BioChat is a production of ABclonal Technology, hosted and edited by myself, Ken Lung. Please rate and review this podcast on Apple Podcasts. Follow us on social media. You can find us at abclonal.com/biochat. And you can, of course, find our various socials in the show notes linked to Dr. Beaker's page on ABclonal.com. We will also have the YouTube link for this in the podcast, and also the podcast link for this in the YouTube link. So no matter what, you'll be able to find us. If you wish to contact the podcast directly for an interview opportunity or any comments, or to inquire about ABclonal's quality products and services, please send a message to service@abclonal.com. Thank you for watching, thank you for listening, and we will see you on the next episode.